Good evening, everyone, and welcome to the Pubcast, where leading scientists and industry professionals meet over a few drinks to discuss the latest ideas and trends in animal nutrition. We're here at the 2024 Tri-State Nutrition Conference, where Balchem just presented and sponsored a symposia titled New Discussions in Amino Acid Nutrition. Speakers on the symposia today was Dr. Mike Van Amburgh from Cornell. Topic that he covered was amino acid nutrition for maximizing milk component yield, followed by Dr. Mark Hanigan from Virginia Tech. His topic was titled Understanding Amino Acid Bioavailability. Then we followed that up with Dr. Chan Lee from The Ohio State University. His topic was Current Understandings of Lysine Nutrition in Dairy Cattle. And then last was Dr. Alex Hristov from Penn State University. And his topic was Histidine, a limiting amino acid for dairy cows. And so if you'd like to listen to these, so if you're listening to the podcast, these presentations are now available at balchem.com/realsciencemedia. So why don't we get started, Mike? We'll jump right in with you since you were our leadoff hitter. Give us just kind of a brief treetop level idea of what your presentation was about today.
New research is changing everything we thought we knew about choline's impact on the cow and her calf, and top scientists have a lot to say about it. They are presenting new research that supports choline as a required nutrient to optimize milk production, choline as a required nutrient to support a healthy transition, choline as a required nutrient to improve calf health and growth, and choline as a required nutrient to increase colostrum quantity. This new research is solidifying choline's role as a required nutrient for essentially every cow, regardless of health status, milk production level, or body condition score. Learn more about the science that is changing the game and the choline source that is making it happen. Reassure precision-release choline from Balchem. Visit balchem.com/scientistsay to learn more.
Thanks, Scott. I think the high level concepts were we need, generally we tend to overfeed nitrogen to cows right now, and that we need to feed less, and that looking at nitrogen efficiency in the traditional way may be difficult to allow us to move that. So we probably need to look right to what she's excreting and then use that as our balancing tool. Look at urinary nitrogen excretion, and then balance that out with rumen ammonia. And then once you get that figured out, then you can move right to amino acids.
So for us novices out there, can you better describe maybe what nitrogen efficiency is exactly, Mike?
Historically, we look at nitrogen efficiency as basically the aggregate efficiency of amino acid use in cows. We generally tend to talk about intake nitrogen, which includes all the non-protein nitrogen, right? And then run that against milk or milk nitrogen, right? And I think that confounds it a little bit because in a monogastric, we would be looking at amino acid nitrogen to tissue output or eggs or whatever it is you're trying to do in a chicken or a pig. Whereas in cows, we tend to incorporate all nitrogen, and I think that confounds that a little bit because if you look at the aggregate amino acid values in most of our high-producing cows, they're actually running about 70 to 73% efficiency on an aggregate level, right? Which is, pretty good. But when we work that up to total intake nitrogen, then it gets confounded, and now we're down in that 30-35 range, right? Which is a little unfair to the cow, right? So this comes down our biggest problem in terms of what we do in the field is how do we reconcile those ruminal nitrogen requirements to a point where we can optimize the capture of recycled nitrogen and reduce the amount of nitrogen that's being excreted in the urine.
All right. Dr. Hanigan, you were second on the list. Can you kind of give us a brief overview of what your presentation was all about?
Well, as Mike pointed out, obviously the cow has amino acid requirements. We have some knowledge of those. And the first starting point is, well, what is the supply from our diet? And that's, been an issue really relative to the ruminant industry, relative to the monogastric industry, because they can feed the animals directly, look at, diet minus feces or diet minus ileal, and get a pretty good estimate. We've got bigger challenges because we have to sort of account for the ruminal losses. And so, I basically just tried to summarize that there's, at least 4 valid methods of measuring this. And the issue is not really validity of those methods, it's more the precision of those methods. And so they don't all have the same precision. And one of the classical ones we've used is sort of intestinal disappearance, and that has very low precision. We can't really get enough numbers to be able to use that. And so other ones that, have higher precision, would be the ones that rely on dilution of a marker or a label in blood or in milk. And those actually can allow more animal numbers as well that can get our precision up just from mass volume.
Right. And you were kind of talking about the method that you guys have put together. Would you mind talking just a little bit about that and how you work that?
Yeah, and it doesn't really originate with us. We modified it and made it sort of more usable. But it's the idea that if you take a labeled amino acid, so these are stable isotope labeled amino acids labeled with carbon-13, so there's no a risk to animal health or your health or anything else. And if you include those in the diet or into the animal, not in the diet, because we still have that rumen problem, but if you can either infuse those into the intestine or into the blood at a constant rate, well, then you set up a scenario where if the supply from the diet changes, that ratio of labeled to unlabeled will change, and it changes inversely, right? More from the diet will reduce, will lower the ratio of labeled to unlabeled. Less from the diet will increase that. And that's the concept behind it. And then it's just a matter of trying to, there's some complexity because it takes quite a while for that to all equilibrate with the whole body. And that's the big problem, right? We can't afford to feed the cows that isotope for 40 days to get them to steady state, right? And so there's a bunch of tricks that we use to try to get it done in a much more expedient time.
Mm-hmm. One of the things that you said that I took note of is that, and a lot of your talk was around rumen-protected amino acids and your work in evaluating the bioavailability of those, and you made the comment that some of the actual bioavailability is quite different than perhaps what the manufacturer is saying. If a nutritionist was wanting to know what is a bioavailability of a product that they're using or feedstuffs that they're using, what would you recommend to them?
Well, I'm hoping that, most of the producers of these products actually are doing assessments, right? With one of these valid methods. And if you provide the data to the end user, then they should be able to make a decision based on that. Now, one would always question, okay, was it done properly? And I think it's up to the end user to look into that and make sure that it was done properly and to assess whether or not they think the data is good. We haven't really done a lot of comparisons among products because that's a good way to not have any friends at all at the end. Okay. We have evaluated a lot of products and we've provided a lot of numbers with as best precision as we can. Most of the ones we've looked at, honestly, are prototypes. They're not products on the market. We're trying to help make better products.
Yeah, makes sense.
I have a question for Mike, actually.
Oh.
So there was a slide up there I had not seen before today, the herd in northern New York. That was quite interesting. The when you introduce the Plenish beans into the herd. So there were 3 columns of numbers under both the fat percent and the protein percent. What was the third column represent?
That was the genomic Holsteins.
Okay.
Those were all bulk tanks.
Wow.
Yep.
Amazing.
Interesting, isn't it?
Yeah.
Scary too.
Yeah. What was the butterfat? It was
5.3.
5. Yeah.
Yeah. But you know, I walked to the back of the room. The nutritionist has now left, but I walked to the back of the room and he was telling me about a herd in Western New York that he works with, and the herd's averaging 5% butterfat right now, right? Which means that, you know, some of those cows are 5'6", 5'7", 5'8", or 6, right? So where these cows are at right now and their capacity to make butterfat is incredible. The same capacity exists for protein, right? We just haven't, for whatever reason, we're not tripping that trigger the right way yet. And I don't know what it is that we need to do to get the cows to push up the protein the same way we're getting the fat. All I can think of is it's more energetically efficient to just add a couple carbons to the end of that carbon chain and keep moving than it is to do all the protein synthesis for the casein. But yeah, there are herds out there doing remarkable things right now, especially if they've been on IVF for a long time, right, and really shorten that generation interval.
Right.
You know, I used to make jokes about Jerseys being like water buffalo. I'm beginning to think we can do Holsteins and make them look like water buffalo if we just figured it out.
We're going to go with Chan. Why don't you kind of give us a real quick overview of what your presentation was about today?
So today I talked about potential factors that may cause the variable responses to feeding rumen-protected lysine. It was not just lysine, it's probably about the rumen-protected methionine and probably histidine. What I talked about is there, as Dr. Hanigan said, if we delivered the right amount of amino acid and increase it, cows will respond. However, if you look at the individual studies and between studies, there are something that we we have to understand to increase the response, especially rumen-protected lysine. If you look at the individual studies for rumen-protected lysine, the responses are not as good as rumen-protected methionine or rumen-protected histidine. So what I have been thinking is, so now we developed the good models. I'm pretty sure this is very good models. But we all know that those requirements of amino acid were developed based on protein building blocks. But maybe we are missing something from that concept because there are a lot of different roles of amino acid other than protein building blocks. So what I suggested or what I included in my presentation, understanding those roles. I'm not sure if we can understand everything, 100% of individual amino acid about different roles other than protein building block. But next amino acid research, we may need to spend time and put our effort to understand more about amino acid roles other than protein building blocks. So next 10 years later, when it's time to update our models, maybe if possible, we can include those information in the model. So then I'm pretty sure that we can increase the response to feeding rumen-protected amino acid or additional supply of certain amino acid, and we can make that response more consistent. That's what I mainly talked about in my presentation.
Now, one of the questions I had as I was looking at your data is that a lot of those studies were 10, 15 years old. And I was curious if we had adequately characterized the bioavailability of those products going into that study, and if that did not contribute to some of the variations in responses. Do you have any thoughts relative to that?
I think what we don't know, I'm pretty sure so there are many papers available about the differences between in vitro and in vivo papers, bioavailabilities. And what we miss in this about the bioavailability of rumen-protected amino acid is once we have a bioavailability from a certain method, we use it for all the time. For early lactation cows, mid lactation cows, or any diet, basal diet. But I think depending on high forage diet, high starch diet, or early lactation cows, because they have a low intake, passage rate will be different. Bioavailability might be different between, even this is the same product, bioavailability may vary. I think that's another area that we have to do the research in the future.
All right. Very well.
I think maybe to add to that too, and I think maybe Mark brought this up in at the end of his presentation. Maybe it was in yours too, Chan, but the whole feed stability aspect is it's pretty overlooked right now, right? There hasn't been much research looking at that, but I think that may be playing a role here as well.
So, that certainly could add to the variability among studies, right? I mean, you could all use the same product, and if it was handled differently, it could end up with different results. Okay. The other thing that concerns me a little bit, and I understand sort of that we need to feed these feed in the end, right? So we got to make decisions, and it may not be on the best knowledge, but I think one of the things we need to really be careful with in these meta-analysis is if we just look at, for example, one amino acid, and I think, Chan, you showed that lysine, that you had 3 or 4 studies that had dry matter intake responses. Well, as soon as dry matter intake changes, all bets are off. Okay, that's not an isolated study anymore. You either can't use it, or you got to account for all the energy and all the amino acid changes. And so I'm a little bit worried that we might have some variation, even in what we say as our numbers, just because of the trying to pull all this disparate data together to get something out of it. But no doubt there's a positive response to them. It's just trying to understand, okay, why is it different? One of the things that we did, for the NASM, because, we had a lot of critical people on the committee, rightly so, is, we had to assign values to all those different rumen-protected amino acids if we're going to use them. And if we weren't going to use them, we couldn't get past methionine and lysine and maybe histidine. There just wasn't enough data. And so I had to show some of the other committee members that, okay, we didn't bias our results by what we assigned to those. And the way I did it is we had infusion studies as well, and then we had the non-infused, non-rumen-protected. I showed that the errors, for each one of those subsets were the same, that they weren't biased, okay? So I don't know that I have, in that paper, which is also in press, whether we have all the correct bioavailabilities assigned to all those different amino acids, but it sure looks good on paper, okay?
Yeah, good point. Dr. Hristov, we haven't heard from Penn State yet. Why don't you give us just kind of a brief overview of what your topic was relative to histidine?
Yeah, sure. Well, I talked about histidine, one single amino acid, but I was thinking when you were asking the other speakers here, how slow progress we make in ruminants with amino acids compared to pigs and chicken. I mean, these guys are feeding entirely on amino acid basis, and we are still struggling defining requirements for these 3 amino acids. So my talk was basically about our work with histidine at Penn State, which started with feeding low protein diets for ammonia emissions and environmental reasons. And then we realized that the amino acid that seems to be the most efficient in this kind of diets was histidine. So then we started feeding rumen-protected histidine over a number of years and experiments. I think we have a pretty good data set to kind of define, at least in this range of milk production, milk protein, and dry matter intake, and crude protein content of the diet, what the histidine requirements are. But that was my talk about mostly histidine.
Yeah, thank you for that. One of the things that I found curious, I've often thought that microbial protein was among the best protein you could feed an animal, and yet you tell me that it's very low in histidine. So from an evolutionary perspective, you'd think that would be higher, but maybe can you talk a little bit about that?
No, I have to correct you. It's not very low in histidine.
Okay.
It's simply because we are comparing to methionine, and methionine, because they are metabolized the same way. And we know methionine is an important amino acid in dairy cows. So when you compare these two, there is about, in our data at least, up to 27% lower histidine than methionine. And that also brings all the other responses in terms of metabolism of protein supply and microbial protein proportion of metabolism of protein and so on. So I wouldn't say it's too low. It's just lower than methionine.
Lower than methionine. And yet, I believe, if I heard you correctly, you need them about the same proportions, though, in the diet.
Well, in fact, you need more histidine than methionine because of this deficiency or not deficiency, but lower histidine in microbial protein.
Yeah.
And they are secreted in milk at about the same level. And they are metabolized similarly. So all this together clearly shows that you will have a higher requirement for histidine than methionine.
Okay, very well. You know,
if I could just add a quick comment on the evolutionary side. I mean, we got to remember that cows, up until the last 150 years, when we started selecting for them, really only needed to make 4 or 5 liters of milk. Okay,
Exactly.
That's the most.
Exactly. Yep.
And so we're now asking them to make, 50 or 60 liters of milk. And we can't assume that they would have evolved to this point because we only gave them 100 years to do this. So.
Yeah, no, well said. Makes a lot of sense. And it reminds me of, Mike, what you started out with your presentation with, how cows have changed and how rapidly they're changing. Can you talk a little bit about that?
Sure, Scott. The advent of genomics has really changed everything. And what's exacerbated that is the fact that there's enough money out there now because of the progress they can make where they're now shortening the generation interval through IVF, right? So now we have instead of 4, 5, 6, 7 year generation intervals, we're now down to, a year and a half at best, right? They can get embryos out of a 4-month-old heifer or less. So, with young sires, they're just moving right along at a speed that is way beyond anything we've ever experienced before.
I know it's hard to look into the future, Mike, but do you expect this to continue, accelerate? What the change?
Yeah. The geneticist Wiggins wrote a paper that said they think from their side that they're at the maximum genetic progress right now.
Yeah.
What they're not accounting for is what the dairy producers are doing with all of this embryo technology.
Yeah.
And that's just accelerating it. And it, it looks almost exponential, right? And I don't know what that means.
Interesting.
I think what it means to me is if they were a pig, we'd have a couple different genotypes by now.
Right.
Right. And we, to Alex's point, where they say 3 grams of lysine per mcal of DE, and then they get a new genotype, and then they say 3.1, and everything else is a relationship to that. We've made that kind of jump already since genomics, and which means we really don't know where they might be. Right.
I think that's our biggest problem, right? Is we only are able to get that potential expressed if we can feed them and manage them.
Exactly, Mark.
And we didn't really have a good understanding before, and now they've changed another twofold, and we aren't keeping up.
We are not keeping up.
That's a great discussion. Carrie, I'd like to go to the audience. We got a question out there?
Actually, it was just a comment relative to, so you mentioned the evolutionary limit on the bacterial protein. I thought I saw in the audience Dr. Newbold, who would be wiser to comment than I am. But, when I have talked to other microbiologists about histidine, and as we look at the industry and having to encapsulate histidine and rumen supply, it is a very expensive energetic mechanism for the bacteria to produce histidine. And so one of that limits is why you don't see a high proportion of histidine in the bacterial protein is that it's compared to lysine or some of the others. It's extraordinarily expensive. It's got 3 nitrogens, etc. It's got that extra energetic. So fundamentally, going back to Mark's comment, historically the bacterial profile has never had the level of energy excess to devote to accelerate histidine production. That's why we're going back to fermenters, etc., to produce a commercial product.
Yeah, excellent comment. Thank you for that. Anybody want to expound on that?
No, I mean, it's a good comment. I just think evolutionary, these cows just didn't need that histidine that the modern cow needs or will need if we keep increasing milk production, milk protein, and then at the same time try to feed a low protein diet because of environmental reasons. In terms of producing these amino acids, that's another thing. I was in a meeting last week, and there were pig nutritionists there, and apparently they are start feeding histidine now to pigs, which I didn't know.
Hmm.
But they are going down the list apparently, and now they're including histidine in their synthetic amino acid supplementation. So, in terms of cost to produce it, and then of course it has to be protected. Things may be changing in the future if the pig people and the poultry people feed it.
All right.
Could I ask a question of those two over there?
Absolutely.
So along this histidine thing, so Mark Garner, when he was working with Jim Russell, they came up with this. They identified this bacteria, Alcinella histiformans, that converted histidine to histamine. Right? Is this what do you know? And I have no idea. I still because nobody kept up with this bug. Is this one of the reasons this is a limiting amino acid in a cow? Is because they're converting the histidine to histamines in the rumen, and we just don't get enough flow out of the rumen? Does anybody, you guys got any insight on this? Because it's been something that's been bugging me.
I don't.
Say
Yeah.
So, in one or two of the studies that Susanna did, we looked at histamine, rumen histamine, and there was no change at all. I cannot directly answer your question.
Oh no, but that's good to know.
There was no difference in with supplemented, not supplemented diets in histamine concentration in the rumen.
Indirectly, that probably answers it, right? That's good.
Indirectly.
Well, I mean, it looks like the animal's very careful with its histidine supply. It doesn't waste it. Okay.
No. That's why I wondered if it was the bacteria kind of getting in the way of our efforts. Yeah.
Good question. That's a good discussion and good point there too.
Well, in a side, direction from that is, we tend to look at feeding animals, we have to look at it from a cost basis as well. And so do we focus on the ones that give us the biggest milk protein response? Or do we focus on the ones that give us the biggest economic response, right? Because histidine may be a very good one I think, for doing milk protein, but it's expensive, okay? And so it's not an economically attractive thing right now, unless the poultry and swine people start feeding a lot of it so we can get the cost down. So maybe we should be looking at some other ones just because of economics, not because of, order of requirement in our perceptions.
Yep.
Yeah. Carrie, you got another question out there for us?
Dr. Van Amburgh, you mentioned in your studies you had really good producing cows, getting decent butter fat, but you were able to use amino acids to jump that butter fat quite a bit more. Can you talk to the practicality of how you went about that?
Yeah. Yep. Because in the interest of time, I didn't give you all the stuff that we did. Not that there's any real secrets there. So, the amino acids I laid out. The other things that we did to make that happen is we actually pulled the fatty acids back, and we made a blend of fatty acids. So I'm looking at C16, not C16, C18 or C16, 18.1, 18.2, not overfeeding, because Adam's lock, some of the data coming out of Locke's lab would say if we overfeed the fat, the de novos will decrease, right? The cow, his words are the cow gets lazy. I think it's just energetic efficiency. But we want more de novo and more mixed. So pull back on the fat, don't overfeed the fat, have a blend of fatty acids, increase the sugar and pull back on the starch, right? One of the things that I think is missing in this whole thing, we get focused on acetate and we forget that maybe we need a little bit more butyrate. And if you think about high pasture diets, where you're probably at 2% starch at best and you might be 25% water-soluble carbohydrates, it's completely inverse from what we do with cows here, right? And those cows make plenty of fat, right? With and still have enough propionate to make decent milk. So I think there are a couple things we need to do. Increase the sugar. And, Will Hoover and a bunch of those guys, 25, 30, 40 years ago would always suggest 5 to 7% sugar in our diets. And that's kind of where we will settle out. Pull back on your fatty acids, make sure you get the highest digestible NDF you can possibly get. But, that's a crapshoot on most dairies today because you just don't know what you're going to get. And then bring your amino acids in and don't overfeed the fat.
Mike, just to confirm, were those your steps to optimize energetic efficiency?
That was part of it. Yep. But I went so fast through.
Yeah, I was going to give you an opportunity. If you want to expound on that a little bit more, please feel free.
I just did it.
All right. Very well. Kind of on histidine, one other question I had for Alex is, I was intrigued by some of the research that you've shown, saw positive responses to histidine. I'm kind of curious, how long does that take for that response to happen?
Mm-hmm. Yeah, so I think we estimated that these body reserves of histidine, if you have a histidine deficient diet, hemoglobin, carnosine can supply histidine for up to 7 weeks, but these are extreme estimates. I would say probably no more than 4 weeks before you start seeing drop in blood histidine. So in 4 weeks, you will have enough histidine coming from these histidine depots to maintain blood histidine, and you won't see a deficiency. And that can be a problem in a Latin square type of design experiment.
Right, and I would think in the crossover design as well.
Yeah. Well, crossover is basically Latin square is a crossover.
Yeah.
But that's one unique feature about histidine. There is no other amino acid that has this kind of body reserves.
Okay.
Yeah.
Well, and that maybe explains why, when people do crossovers that are based on protein. So like, the study that Van der Haar published a year or so ago, 1 week. I mean, within 1 week, it was a full response in both directions.
Yep.
And infusion studies that Metcalf published one quite a few years ago, because we had looked at it on that project. Within 3 milkings, we were actually seeing most of the response. So, but it's protein. It's not
protein.
Yeah.
Can I ask a quick question about histidine? So we finished one experiment with histidine. It was different levels of dietary protein diet, supplemented with different amino acid. One of the major amino acid was histidine. We conducted the experiment for 6 weeks, if I remember correctly. We didn't see any difference in dry matter intake. We didn't see any difference in milk yield and milk protein yield. But we found histidine was numerically decreased. It was not significant in the blood. But we found hemoglobin level was decreased. So hemoglobin is very important to carry oxygen to the body, and that is utilized for energy synthesis, ATP. I mean, but is it changing hemoglobin level in the body? Do you think it's going to change the metabolic efficiency of nutrient utilization in the body?
If you ask me, the answer is I don't know. Maybe that drop in hemoglobin you have seen is because it was supplying histidine, or it was used to supply histidine. Although you didn't see differences in plasma histidine, right?
So the hemoglobin level dropped when histidine was deficient. When we provided Rumen-protected histidine, hemoglobin level was higher.
Went up.
Yeah.
So exactly what we have seen as well.
Yeah.
It is possible, Chang-Hee. I just have to say, though, the differences that we see in histidine, in hemoglobin, are very small. They are statistically significant, but the body will maintain hemoglobin levels as much as possible. because it's such an important compound here. So there has got to be some metabolic effect of low hemoglobin.
Yep.
If you, so drawing maybe from another source, if you feed nitrates, right, you can tie up and reduce methane, right?
Yep.
But of course, you get nitrate overspill into blood, and it binds with the hemoglobin and ties up some of the oxygen carrying capacity.
Exactly.
And if I remember right, I mean, those studies were seeing 10, 15% hemoglobin, tie up, and the cows were still performing fine. So it sounds like what you're saying is no, it's not going to be enough to change energy
within this
within that range.
Thank you.
Carrie, any other questions from the audience?
How do you explain amino acid carryover effects?
We never see them.
I do
we
longitudinal
look
studies.
for them and we don't see them. I mean, I think Dr. Hristov's data is very, very compelling, and I buy that for histidine. We've never seen it for any other amino acid. They basically flip over in 3 or 4 days, and nothing seems to be hanging on, creating a carryover effect.
So Alex, given the data you showed on carryover, the applied question is, how long do I wait after starting to feed histidine or other amino acids to see whether I have a response? In a farm situation, what's your response time?
Well, to repeat what Mark said, I think you will see it right away. For methionine, lysine, for histidine, again, if you really are overfeeding, I mean, overfeeding is a rough term, but if you're feeding above requirements, let's say, if we know where the requirements are of metabolism of protein, you probably won't see a response to histidine. So you have to be at or below. And I still think that you will see a response within.
So if you start with an insufficient diet, below requirement for histidine, you think the response will still be quick, still that.
If you start feeding this low protein diet now, of course it will take weeks before those body reserves are exhausted or at least diminished, supplying histidine to maintain blood histidine levels, and then you will start seeing a response. But if you are talking about farm conditions, you are not going to feed that diet from day 0 and start feeding histidine from day 0. You'll be probably feeding this low protein diet for a period of time, and then you are starting supplementing histidine, then you will see a response, I would say, almost right away in a week or a couple of weeks. Yeah.
So what is happening with plasma histidine levels in very early lactation cows?
Good question. Somebody was talking about, Chunky was talking about fresh cows and transition cows. We do have a histidine, I won't call it transition cow experiment going on right now, because it is actually a fresh cow experiment. We started 3 days after calving. These studies are extremely difficult. I didn't want to do them, but I mean, that's pretty much the only thing left with histidine, at least for us to do. So that's why we are doing it. My guess will be, because of the low dry matter intake and of course the high metabolic needs for amino acids, the response will be there. We are at about, I mean, of course we have a 240 cow herd. Until we get all the cows in the trial, it takes months and months, but we are at about 35 cows at this point, and over the peak of the experiment. So soon we'll know, we'll have the results from this.
I think histidine supplementation for fresh cows is more practical way that we can utilize a rumen-protected histidine if the concept about histidine deficiency is correct, which is the microbial protein supply and histidine, low histidine concentration in microbes. So that's what I was thinking about histidine, to maximize the efficiency, effectiveness of providing rumen-protected histidine.
Yes, Changi. The other thing is that we always, I don't, I mean, you may have different opinions, but we tend to feed RUP in this stage of the lactation. So that may mask a little bit the histidine problem.
Yeah.
But generally speaking, you are correct.
Gary, I think we have another question.
On these low protein diets, let's just say 15-5 is low protein diet. And do you feed much of the blood meal or blood meal derivatives in those cases because you're having to make up through all the other amino acids?
So when I say low protein, I mean below 15.5, but 15.5, let's say, is a good target. If you have blood meal and it's a good blood meal and if it's a reasonable price, that's the best source of both lysine and histidine. We don't, because we are trying to monitor the amino acids and balances, but that would be a way to supply histidine.
I said, but in your studies, you're not using any blood meal because of variability of blood meal.
No, not because of variability, because we want to control, let's say, histidine deficiency or histidine supply
by itself.
By itself, not by the other amino acids that come with blood meal.
I'll jump in there. So we've got somewhere we've gone down to 13.5% on crude protein, if you want to use crude protein as your metric. And yes, in those situations, I don't know how to do it without blood meal. Yeah. But and the cows will perform well. We've gotten 45 kilograms of good milk out of about 13.5% crude protein. But to the point that's been made, that the variation around, we conducted one experiment where we had tested the digestibility of the blood meal, thought we had it all worked out. The cows said, nope. I was yelling it at everybody, right? Because I thought something was wrong. They weren't feeding them right. Come to find out that the blood meal switched from the time we tested it to the load we finally got, right? Yeah. And then it corrected itself, right? So we have this beautiful study where, we did it, we got something different, and it came back again. But it's maddening, right? Because you don't want that to happen on a dairy. So, we still feed some blood meal, but we do it in a guarded fashion because I don't know how else to get some of these amino acids when you're trying to bring the true protein down or the nitrogen down far enough. It is a bit of a problem.
I have to say, though, anytime we went down to 14 or below 14%, we have always seen a decrease in production and dry mat intake, milk production and dry mat intake. Maybe we don't have the magic cows that Mike has up there, but
not the cows, Alex. It's not the cows.
Yeah,
they're all the same cows, man.
in our case, and we didn't feed the blood meal, obviously. Yeah. We did see a decrease in production.
Gary, I thought I'd seen a hand over here at one point in time. I don't know if they've left or not. Any questions over there?
Hello. Back in the day, we used to be trying to get spike in milk production from supplementation at a 16%, or what would have been a normal percent protein. And the place we got it was not from lysine or histidine, it was from methionine. Now, are we just working with a different animal? Not a different animal, excuse me for even having suggested that. A different scenario when we're looking at these very low protein diets. Do you get nothing from methionine when you're feeding a 14.5% protein diet?
No, to do that, Tim, that's a great question. To do that in my book, we're feeding probably somewhere between, if the cows are consuming 70 to 72 mcals of energy, we're somewhere in the 80 to 82, maybe 84 grams of metabolizable methionine. So methionine is what you're hearing from me is that I already took care of the methionine. That's out of the way because that one's easy. We know how to deliver it. We know the bioavailabilities of most of the ingredients. We just move ahead. Now we've got to the discussion here. Now you've got lysine and histidine where we're still kind of trying to figure out what we're going to deliver and how much we got to go.
Very well. So I think I'm going to call last call, gentlemen. I usually wait until I'm done with my bourbon, but I got a triple this time, and so I'm not
I was
going to
going to
finish
ask
it.
you about that.
That was quite hefty, wasn't it?
Yeah, a little bit.
Well, I restrained myself. What I'd like to do is ask each of you to kind of give us one key takeaway, maybe something you learned, or maybe a piece of advice that you'd like to impart to our audience here today. And Clay, I'm going to start with you, if you don't mind.
Our last call question is sponsored by AminoSure XM Precision Release Methionine, the next generation in amino acid balancing. With AminoSure XM, you can save up to 5 cents per cow per day on your methionine investment. Try it today and receive an additional 2.5 cents per cow per day savings with Balchem's limited time rebate offer. Contact your Balchem representative to learn more.
Sure. Yeah. So obviously, amino acid supplementation is very critical as the genetics are changing on these cows. We're really seeing some pretty amazing responses, partially related to amino acid feeding. And I thought all the talks were great, actually, and really blended together quite well. And I think some other key points were, be careful about, where these bioavailability numbers are coming from and how you're handling these products.
Yeah, great comments, Clay. Dr. Lee, I'm going to go with you next.
I agree with Clay. I'm personally interested in bioavailability of rumen-protected amino acid because this is the key information that we can increase the response to feeding rumen-protected amino acid and also make the consistent response to feeding rumen-protected amino acid. So there are many methods available. Dr. Hanigan talked about what we need to look at if we have a bioavailability and what are available. He showed the approach that we have to take to get a good information, have a right information from about rumen-protected amino acid.
Yeah, great comments. Dr. Hristov?
Yeah, I would say that nitrogen is coming back. We've been all crazy about methane, at least in my world, in the last years. But nitrogen has been there, is here, and it will come back. And if nitrogen is here, we'll always be talking about amino acids in animal nutrition, and that will just continue. Hopefully, we'll make even a fraction of the progress that the monogastric people have made in the next few years.
Thank you. Mark, what kind of comments do you have?
Yeah, I think as we move forward, from an industry and particularly a science standpoint, we continue to discover, new things, right? But sometimes we get caught in this trap of thinking, okay, well, I have to maximize milk protein, or I have to maximize microbial protein, or I have to find the best amino acid, it has to have the highest bioavailability. All of these have economic costs, right, for industry application. And so the best may not be, and often isn't, the most economical decision. It's somewhere off of the best. And so I would challenge, the idea that we have a 45% limit on nitrogen efficiency. I think it's higher than that because we are not allowing ourselves to say, okay, we're going to accept lower microbial output, and then we're going to make it up with RUP or amino acids or whatever. If we knock that off the table, we never are going to get beyond that boundary.
Excellent comments. Mike?
Yes.
Bring it home for us.
Yeah, well, what Alex said and what Mark just said, I fully agree with all of that. I think where we're at right now, and to play off of what they just said, the cows are changing. We need to figure out how to challenge ourselves to work to understand what our cows have the capability of. And that's going to play into everything that everybody just said here, right? We're going to have to kind of take off our blinders and say, hey, it isn't just one thing, but we have to work pretty hard to sort it out, because it will have to be economic. I think the question, to Mark's comment, and I struggle with this, Mark, what is the economic response, but what is the biological potential, right? And I think we got to go figure out what the biological potential is, and then decide, is that economically viable or not, right? Because right now, our milk protein price right now sucks, right? So there's no incentive to make more milk protein. But we can make all the milk fat we possibly can, right? And it doesn't seem hard to do that because we can't figure out how to move the protein. So I think we're going the right direction, but we got to keep challenging ourselves to figure out how to do that.
Right. And so we have to have that full response surface for all
Exactly right.
all those relationships, right?
That's right.
So we can move up and down it as we need to, because tomorrow the market's different and the next day it's different.
That's right.
Yeah.
Exactly.
Yep.
Excellent comments, gentlemen. I want to thank you guys for joining us today. It's been a great symposia and a great podcast. To the live audience, I want to thank you for joining us for our 100th episode. And so if you'll join me in a quick drink, I would greatly appreciate it. Thank you. Cheers.
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 And I'd like to thank our loyal listeners for joining in once again. We hope you learned something. We hope you had some fun. And we hope to see you next time here at the Real Science Exchange, where it's always happy hour and you're always among friends.
We'd love to hear your comments or ideas for topics and guests. So please reach out via email to anh.marketing@balchem.com with any suggestions, and we'll work hard to add them to the schedule. Don't forget to leave a 5-star rating on your way out. You can request your Real Science Exchange t-shirt in just a few easy steps. Just like or subscribe to the Real Science Exchange and send us a screenshot along with your address and t-shirt size to anh.marketing@balchem.com. Balchem's Real Science Lecture Series of webinars continues with ruminant-focused topics on the first Tuesday of every month, monogastric-focused topics on the second Tuesday of each month, and quarterly topics for the companion animal segment. Visit balchem.com/realscience to see the latest schedule and to register for upcoming webinars.